speaker, Dr. Tony Berardino from Canada. She is the scientific director of astec-biology, and Tony has over 25 years of experience in directing IVF laboratories. Her main interests are in embryo culture system and vitrification. Please, Tony.
Thank you. So thank you very much for this kind invitation, and thank you again for the people that stayed this late the last session to listen to our talk. My talk is about culture environment and its potential effect on embryo development, and really looking at comparative analysis of incubators. My disclosure is that I'm the scientific director for astec-bio. So I think it's very important, and this is definitely an old slide, but it's a slide that I like to present all the time, and it's about laboratory factors that can actually affect the outcomes of IVF. Don Breakers in 2015, together with Jason Swain and Rusty Poole, they looked at the factors that would impact every single procedure that we carry out in IVF. And at that time, they identified 175 factors that would actually come into play when we want to have a positive outcome. Since then, this list has been revised, and right now we actually think that it's going to be over 200 factors, only because our job in the lab got busier than ever. And so when we look at that and we look at that list, we want to know what are the factors that actually affect embryos. And in this case, we're looking at lights and pollutants and oxygens and everything else that we actually have in the IVF lab. And therefore, minimizing environmental induced stress is really crucial in the IVF lab, because what we want to do is really have a culture system that optimizes embryo development and eventually achieve the highest possible reproductive outcomes. So when we look at incubators in the IVF lab, we know that they play a pivotal role in providing a stable and appropriate culture environment that is required for optimizing embryo development and clinical outcome. Incubators regulate key important variables. We all know about gas concentration, temperature, humidity, pH, osmolarity. Those are all factors that really play an important role in culture systems. And importantly, these potential stressors can impact the laboratory incubator. As a result, the incubator is arguably the most important piece of equipment that you will have in your laboratory. They control multiple environmental variables, and really, that's where your embryos sit the longest in the IVF lab. That is, we all culture embryos now to day 5, day 6, day 7. And so the incubator becomes a really important piece of equipment in your laboratory. And so we want to make sure that incubator selection and management are critical, again, for success. But I think one of the things that we don't realize, especially nowadays, certainly not when I first started, we had one big box incubator that came from growing cells in the research lab. But right now, what we have is a great selection of incubators, and really it's management, and when you're purchasing, it becomes a complex process, due mainly to the technology advantages and sort of varying capabilities of incubators. This is a slide from Jason Swain's paper back a few years ago, but he wanted to point out here that when we're comparing the selection of incubator, when we're trying to think of what incubators we want to put in our laboratories, there are many factors that you need to look at it. What kind of gas types do you want? Do you want just CO2? Do you want premix? Do you want a low oxygen? Also, I think it's very important that you sort of realize that the CO2 sensors and O2 sensors in incubators are not the same. So really, you need to have an informative decision making when you're picking up incubators, because those are not the same. Also your temperature controls, those are very important things. The design, do we want a big box incubator? Do we want a benchtop incubator? You know, most labs, we organically grow and we kind of keep old incubators in the lab and then we have some new ones. And like, do we have to have them all the same or can they be a mixture? Again, humidity. I think a lot of people have read quite a bit about humidity. Do we want a dry incubator? Do we want a humidified incubators? And what does that look like? And again, other features. So the point of the slide is really sort of managing your incubators and when you're looking at one. I think also very important is the fact that if you're trying to look at comparative studies and clinical outcomes, the data is very, very limited. Very few studies actually compare the performance of different incubators in the laboratory. I think once time-lapse came around, we started comparing incubators to time-lapse, but we have to realize that those are two different beasts, right? They're very different in that. Few studies really report on environmental stability of an incubator and their clinical outcomes. And of course, there are quite a few confounding variables, such as air quality, embryo end weight, that complicates really the assessment of the incubator. So the study question was to really study the effect of incubator management on assisted reproductive outcomes and compare clinical data that's been published in standard competency and benchmark values. So the study design was this is a retrospective data of IVF cycles, IVF and ICSI cycle performed in a private clinic in the UK. And the data was from January 2024 to June 2025. So a good length of time. They looked at different parameters of embryo development, and really they compared it to the Vienna consent report. And I think most of you in the room are familiar with the Vienna Consents Report. The Vienna Consents Report really looks at minimum benchmarks, both as competency limits and aspirational goal benchmarks of what we should look at. So the study design was that all sites and patients with primary and secondary infertility were allocated to an AC-10 incubator. This is an Astec incubator. They carried on insemination both with conventional IVF or ICSI. Embryo culture was carried out in their normal protocols. And then what they did was they looked at the value compared to the Vienna consents forms. So when we looked at the results, they looked at fertilization rate with IVF, fertilization rate with ICSI, blastocyst formation, whether there was an ICSI degeneration rate, and also they looked at the frozen blastocyst survival rate. The first column that you're looking at is the clinic's results, and then they looked at both the competency values from the Vienna report and the benchmark values from the Vienna report. And really you can see here that when we look at the p-value, there was no difference in those results. They were very much within the two parameters of both the competency and the benchmark. And so when we look at the result, we found that there was no significant difference between the clinic's results and the published competency and benchmark values in the Vienna Consents Report. And really the adoption of EC10 incubator provided by Astec was a very dependable incubator that provided stable temperature and CO2 control. Of course, the limitation of the study is that each patient was its own control in the group, as well as for their oocytes and embryos, because they were all cultured in the same incubator. And we really did not have a cumulative pregnancy rate or implantation rate, and I think this, and obviously birth rate, and this is really confirms that more studies need to be done to confirm that really that difference is not there. So the conclusion of this study was that proper management of incubator, including volume, workflow, is paramount in optimizing the function of an incubator. Clinical data obtained from culturing both oocytes and blastocysts in an EC showed no statistical difference when compared to the Vienna report. And incubators in the IVF laboratory really plays a pivotal role in providing a stable and appropriate culturing environment requiring for optimizing the outcomes. And so much care is needed in order to, when you're purchasing one. Thank you.